Hello and welcome to my presentation on the 7th topic of molecular biology, which will be DNA replication, transcription and translation. And just a heads up, in a short amount of time I'll also be uploading a video on the high-level topic of nucleic acids, which delves much deeper into the topic of replication, transcription and translation, with all of the different enzymes that are involved in it. So within this video I'll be having more of a surface level analysis of the processes mentioned here, but if you want to know it a bit more specific, wait for the video on the 7th high-level topic, nucleic acids. Again, I'll not be delving into much detail here, but only the most important facts for those three processes of biosynthesis. So yeah, without further ado, I hope you enjoy, and it's self-explanatory. I'll be talking about replication, transcription and translation. So yes, DNA replication is important, as the DNA must be able to replicate itself accurately so that the genetic code is passed on to the daughter cells during cell division. And this takes place in the nucleus during the interphase, the so-called synthesis phase of the interphase in the cell cycle, where the DNA is not tightly coiled. And if you're interested on more of that, check out the topic of cell division and cell biology that I made. Shameless plug. So DNA replication is semi-conservative, which is important. So pre-existing strands act as templates for newly synthesized strands. So one of them remains and the other one is newly synthesized, which is why, yeah, it's semi-conservative. And here you have the basics of the replication. You can part it in three ways: the unwinding of the DNA, or the unzipping at the replication fork with helicase, where it's untwisted and separated, as you can see here, and coated and prevented to rewind again. And this happens at the replication fork that you can see here. And then the second part is the synthesis of the new DNA strands, where first you have RNA, short stretches of RNA nucleic acids that are complementary to the template, where then another enzyme comes in on the two strands and assembles DNA nucleotides on one strand that happens continuously, which is the leading strand, and it happens in short fragments, so-called Okazaki fragments, and this is then the lagging strand. That is because DNA replication goes from a 5' to a 3' direction, and because, as you remember from the structure of the last video, they're anti-parallel, so one goes 5', 3', the other one goes 3', 5'. So that is why one of them is leading and one of them is lagging. And the third one would be the rewinding, so the protein removed. So we have enzymes that remove the RNA and they are replaced with DNA, and these strands are sealed and they become two different ones. So we have two new made from one. And this was also showed in the so-called Mendelssohn and Stahl's experiment that showed the DNA replication to be semi-conservative. But I'll be delving into much more detail in my other video. We also have a so-called polymerase chain reaction that is the artificial method of replicating DNA in the laboratory. And the first step is the denaturation, because if you remember, enzymes are proteins and proteins may denaturate, so lose their biological functions and unfold, for example with temperature, which is what happens here. So the first denaturation happens when the strands are separated with a heightened temperature of 90 degrees. The second step is the annealing, where primers are annealed to it, and that happens at a temperature of 55 degrees, so still above the denaturation point, but lower than the denaturation point, lower than the denaturation in the first step. And then we also have in the last step the elongation, so the actual synthesis of the replicated DNA. where the sample is heated to the optimal temperature for the heat-tolerant polymerase that's derived from a bacteria, the so-called TAQ, the TAQ polymerase, to function. That functions as DNA polymerase during replication. That happens in a temperature of 75 degrees. Yeah, moving on to transcription. It's the first part of protein synthesis, and it also goes from 5' to 3'. And I believe all of those processes are from 5' to 3', so that's somewhat easy to remember, I guess. And it takes place in the nucleus for eukaryotes and in the cytoplasm for prokaryotes, because, you know, prokaryotes don't have nuclei, sorry. And it's the production of an intermediate molecule that carries a coded message from the DNA from the nucleus into the cytoplasm, where the protein can be produced in translation. So the process by which an RNA sequence is produced from a DNA template, and the intermediate molecule is the messenger RNA, or the mRNA. Their genetic information becomes movable and can be transported from the DNA to the ribosomes for protein biosynthesis, and the mRNA differs as an RNA from DNA, which I've mentioned in the structure in the last video. And the transcription, as you can see in the graphic, in the picture here, takes place in maybe 3 steps, if you want to see it that way. So first, there are special DNA regulator sequences called promoters that are located in front of the affected DNA segment as a starting point and indication of the direction for transcription. And then you have this enzyme, the RNA polymerase, that separates the DNA strands and synthesizes a complementary RNA copy to one of the DNA strands. So you have an RNA copy to the strand. The second part would be that 3 nucleotides align opposite their exposed base partner, and the polymerase assembles those, and then single strand of mRNA is formed, so we have the copy of the gene. And in the last step, the enzyme RNA polymerase removes the additional phosphate groups and uses energy to from the cleavage to covalently join the nucleotides to the sequence. Then the enzyme detaches and the double helix reforms, and the mRNA moves via the pores in the nuclear envelope to the cytoplasm. And I forgot to mention that, but the transcribed strand is the antisense, which is complementary to the RNA, or the mRNA. And the non-transcribed strand is the so-called sense strand that is identical to the mRNA. So yeah, that's transcription. Moving on to translation. It's the process by which the coded information in the mRNA strands is used to construct polypeptide chains, leading to functioning proteins. And this is carried out in the cytoplasm by ribosomes that have their own rRNA, so ribosome RNA, and transfer RNA, so tRNA. And ribosomes have binding sites for mRNA and tRNA, and they consist of 2 subunits of different sizes, so the small subunit and the large subunit. And this one you can also kind of differentiate in 3 different parts. So first, again, the ribosomal subunits move from 5' to 3', and they bind at the start codon of the mRNA, which is most often the AUG, the AUG of the mRNA. That's the sequence most often. And it makes those complementary base pairings between the codon and the anticodon of the tRNA, because the tRNA is basically the mediator between mRNA and peptide. Again, I'll delve into much more detail in my other video. And the tRNA binds to the P sites of the ribosomes. And in the second part, the 2 tRNA molecules in place on the ribosomes form these peptide bonds between the 2 amino acids, and this forms the dipeptide. And then the first tRNA detaches and the ribosome moves along the mRNA in this 5' to 3' movement, one triplet to the next codon, and that goes on to the entirety of the mRNA that needs to be translated. And the third part is the elongation that continues until the ribosome reaches the stop codon, and there is no corresponding tRNA to the stop codon, so the translation termination begins, and the complex of ribosome and RNA separates, and the synthesized amino acid chain is released. And that's basically the process of translation. There's also this hypothesis made in the 1940s that each gene is responsible for the production of one protein. So later it was modified that one gene makes one polypeptide because proteins have multiple polypeptides. And that is also to some extent today's agreement, but there are exceptions that are important. For example, some genes are for regulators and not transcriptional translation, and some code for tRNA and mRNA, which are also not translated transcript, and in a few genes there is a differential expression of certain genes depending on the tissue. So yes, this was an overview of DNA replication, transcription, and translation. I hope you enjoy. These are my sources. Have a nice day.